my pleasure to be here. It's actually the first time that I present at INU. And I apologize for the delay in me and my plane being delayed. So I really appreciate your flexibility. So I'm a statistician by training, but I have worked with biologists since I started doing research. And you will see that the questions that we're trying to solve always originate from a biological question. My talk is going to be very applied, but I hope it's going to give you an idea of the types of problems we're trying to tackle in computational biology. There's a lot of room for improvement in terms of methodological developments as well. So the title of my talk is how do we deal with time course data? So experiments that have been measured at different time points and when they are multi-omics. So multi-omics, I don't know if you are familiar with that term. I don't know how to, okay. Sorry, I should start with the acknowledgement. So there's quite a few students and collaborators who've been working with me, and I'm going to present their work. So Yasmin did her PhD with me a while ago, and she developed a framework based on linear mixed model splines. And then there's been Antoine, who's more on the bioinformatics side from Quebec. And he's been working with a biologist who's my collaborator, Olivier, in integrating different types of data measured on time course. And I have Sarita, who's now a postdoc in my lab, and she's working more on the statistical developments for those type of data. So let me start first with what multi-omics data are, and this is just a little cartoon about where we are. So pre-years 2000, we were in this conventional molecular biologist biology approach, where you measure the expression of a gene, of a parameter, or we call it a variable if you want, across individuals. And you're trying to work out whether that gene has an effect or is affected between people who have a disease and don't have a disease. Okay, so you can think about that as a t-test, right, on one single variable, so the expression of a gene. You can do the same if you measure the abundance of a protein or a metabolite or the abundance of a bacteria. So that's the more very reductionist type of approach. In the year 2000s, we started to be high throughput. And so now, instead of having one vector, I'm having to deal with matrices. So I have a matrix of different types of data using RNA sequencing technologies. So for example, transcriptomics is you can measure the expression of every possible gene. So we have about 20,000 of them. You can do the same. You can use another technology and you can measure the proteins, and you can do the same for metabolites and so on, and bacteria. And so you end up with different types of experiments, all generating large amount of data. So I'm talking about 10,000 to 100,000 of variables, and a very small number of individuals, 50, if you're lucky. And the multi-omic field is quite recent now, maybe 10 years ago, where we are able to do those assays on the same individuals. So I can take your blood, or I can take a sample, and I can measure all the proteins you have, all the genes you have, using all these different types of technologies. And in multi-omics research, what we're trying to do is we're trying to understand the associations or the correlations within a particular type of omic. I would like to understand what are the genes that are correlated with each other, because they may explain specific biological mechanisms. But we're also interested to explain the correlation of the associations between the different layers of omics. Is there a gene for which the expression is highly correlated with a protein? And is it going to tell me that that gene is actually is responsible for the translation of that protein? So this is all data-driven, those approaches. But of course, biologist friends have also a lot of knowledge. And so they can guide you and say, well, actually, you know what, we should have that gene that would code for that protein. So why can't you find it? And so a lot of my work is to actually mine those data and to reduce the dimension of those data to something we can understand. And so going from a holistic approach to a reductionist approach, where we can then validate those experiments, because of course we can't validate 20,000 genes, but we can, if we can identify 1 or 2 genes that may explain a specific disease or the effect of from a treatment, then we can try to validate that. So that's just the introduction. So the first part of my talk, I'm just going to talk about one single omic, just to make things simpler. I don't know. So I'm going to talk about microbiome data. So the study of the bacteria in a particular body site. And I'm going to talk about those experiments that are measured across different time points. So this is an example here where we have the abundance of, each line is the abundance of a given bacteria across different individuals. So you can see here we have different groups of individuals. Usually you have a control and a treatment group. And you can see that sometimes for a given bacteria, there's no group effect, there's no time effect. So the time is on the x-axis and the abundance of the bacteria is on the y-axis. You can see that there is a group effect, or there is a time effect, or there is a group and time effect, or there is an interaction. Okay, so this is a very simple case scenario that we call differential analysis, where you can use, in the context of microbiome data, there's a lot of zeros. So you will see a lot of models based on zero inflated regression. What we chose in our research is to use linear mixed model splines. So I believe that this kind of question has been, addressed quite a lot in the literature, whether it's on microbiome, whether it's on transcriptome or anything. That's something I think as statistician we are able to do. The second problem is a clustering issue. We're not interested in clustering the individuals. We're interested in clustering the bacteria. So this line here, this is a bit different. The line here is the expression of a different taxa and across time. And so basically we're trying to understand, are there bacteria that behave the same across this disease progression? And can we cluster those profiles? And for that, you have a lot of clustering methods that you can use. So we looked in the literature in the context of microbiome data, especially. You have, time clustering or dynamic time warping. The approach we propose, and I'm going to talk about, is using a simple principal component analysis or partial least square regression analysis, combining that with splines. So I will explain to you how we do that. And the third problem that is exciting, but I don't think has been really solved, is network modeling across time. So here, each node here represents a taxa or bacteria. And I would like to understand how are they correlated with each other across time? Or is the network, so the way I see it, when I talk to my postdoc, I say those bacteria are all friends with each other in a given, in a given environment, perturb the environment a little bit, and suddenly they make new friends and they make enemies and they break the relationships that they had with each other. Can we actually capture this in a network? Can we model this as a network? So that's the third question that I think is quite challenging to solve because it's across time. So the differential analysis that I talked to you about, it's a univariate methods. And as I said, there's heaps of them now. You can have a look at if you're interested in those kind of challenges, you can have a look at our review where we actually applied and tested those methods on simulated and real data. If you're really interested in that topic, you need to think about whether you want to start with count data, so the sequencing row count data, or if you are actually normalizing, scaling your data so that they are continuous, because of course you're going to choose different models. For the clustering analysis, this is more a multivariate approach. I think in that field, the problem is that we don't have a lot of time points. All those experiments are extremely costly. They're between $200 to $1,000 per sample per time point. That's why we never have a lot of samples. And also, when you're interested in clustering those profiles across time, you have to expect some sort of regular trend happening. You can't have interventions that really destroy the gut microbiome, for example. So anything with antibiotic, for example, is going to completely perturb your microbiomes, and so that makes things difficult for clustering. And for the network modeling, as I said, there's not many approaches. What we found is that people tend to model or fit a network either on a single individual and try to work out how the bacteria are linked together, or they try to pool all these individuals. And there's a high individual variability in those data. So that's why I think that question hasn't been fully solved yet. It's because there's way too much noise in those data. So that's it for the introduction and the kind of questions you may ask from time course omics data. I'm going to start first by talking about linear mixed model splines for single omics. So again, we imagine we are in that framework. We have a microbiome data set. We have measured the abundance of the bacteria across different individuals. And we want to do some sort of differential analysis here. So in fact, you can represent the data like this. It's a 3D because you have the time component, you have the number of bacteria or taxa you're measuring. So here we're about 500 to 1,000. And then you have the samples, as I said, if you have 50 individuals and they're all divided into two groups, so usually you may have 25 sick and 25 not sick. That's the size you should expect. The problem is, of course, that the sampling is irregular. Often when we work in microbiome, in gut microbiome, we get fecal samples. People don't often send their samples, so sometimes they miss out the time point. And the number of time points is small, and the individual variability is high. So just in general, in omics, it's just high because we're all different and we're measuring something very precise from a molecular perspective. But you can imagine in the microbiome, it's even worse because we have different diets. We didn't even eat the same thing at lunch today. For example, I'm vegetarian, so I'm sure my gut microbiome is much different from those who eat meat. And so that creates a lot of challenges. So one approach was to say, well, we need to reduce the dimension somewhere here. And so what if we actually remove the individual dimension? So that assumes that individuals from the same group, for example, the healthy people, you assume that they have a similar microbiome. And so you can collapse that information. So you can do that with a spline. Okay, so these dots here. The timer is on the x-axis. On the y-axis, this is the abundance of a given bacteria. And the dots here represent the abundance of the bacteria for a specific person. Okay, so here, this is my cohort of individual at time 0. And so you can just imagine, we can see that roughly that bacteria behaves the same across all these individuals. So why don't we just fit a spline? And then we use the spline as a profile that represents that particular bacteria. So this is how I go from a 3D data to a 2D data, where I would have only the information about the time and the abundance of each bacteria. What is very good with splines is that they reduce noise. They take into account the correlation structure between the time points, because we are across the time course. It's the same people that are measured across time. You can do some interpolation if someone has missed or didn't send a sample. But for those of you who may have worked with splines, you know that you need a large number of time points, and the smoothing parameter is actually really hard to choose. So you may over smooth sometimes. So you don't know how far you should go in the smoothing and the summarization of those time profiles. And so instead we looked at the approach from Verbeiler, who said, well, actually you can use linear mixed models. and has splines from there. And I think what was really attractive is that you are into a linear mixed model framework. So you can model the individual variability. You don't need the smoothing parameters through that framework. And you can choose the model that is best for each bacteria. And then you can do everything that you can do with splines. You can interpolate. Once you have fitted your models, you use those models for any downstream analysis that you want. And because it's a linear mixed model, we can test for time and a group effect. So this is an example of the different types of model you can fit depending on the shape of your profile. So this is an example here. It's just a simple linear model for a specific bacteria. The abundance increases with time. And then you can see here it goes up and down with time and so on. And so you can actually include subject-specific random effects, different types of random intercepts and slopes and so on. So it can be actually quite particular about the type of fit you want for each bacteria. And what was interesting is that we found that the more complex the organism is, the more complex the models become. So if you're looking at cells, for example, this is the number of profiles that we fit with a linear model, and then with a linear model with a subject, this one, sorry, not just the linear mixed model spline. And so you can see here the complexity increases as the organism complexity increases. So as a human, there's a lot of variability and we need more and more complex models. The other thing to note here is we remove a lot of the data here just because it's extremely noisy. It goes all over the place, like you can't fit anything there. And so as we go to human, there's more and more noise. And so we also have to remove a very large chunk of the data. And so this is a framework that we finally came up with. Actually, a lot of the work was in the quality control, filtering the noise before we could even go to the modeling with the splines. And then from there on, you could do any type of analysis you want, because you're back into this 2D matrices input. And so you can then fit any method you want there. So you can cluster with k-means if you want, and you can do some hypothesis testing. So we're quite happy then to fit back into those classical frameworks. Any questions so far? So just to summarize, I just talked about basically fitting a linear mixed model for time course or for longitudinal data. Now I want to move to the multi-omics side of things at a single time point, because before we can go to the integration of multi-omics, I have to explain to you how we, across time, I have to explain to you how we do this part here. And again, it's a lot about dimension reduction using multivariate methods. So we use matrix factorization techniques, and I'm sure you know about principal component analysis, but there's some other methods such as projection to latent structures or PLS that are useful to reduce the dimension of the data. So just for some of you, it might be a recap, depending on how much you know about PCA. But basically here I'm at a single time point. I've measured the expression of genes across samples. This is a big data matrix, difficult to invert. And so instead we factorize this matrix into different vectors. One are going to be called components, and the other ones are going to be called loading vectors. And they are basically low ranks approximation of your original data matrix. Okay, so this is a dimensional reduction technique. What is very nice with this kind of approach is that they are not non-parametric. And so that works well with a lot of biological data, such as microbiome, proteomics that do not have a Gaussian distribution or any norm distribution we know of. And really what we're interested in is to capture the variation of the information from the data. So if I just go to PCA, which is an exploration technique, what I'm trying to do is I'm trying to find the components that are a linear combination of those P features that I have here. So if we are working with gene expression data, you may have 20,000 genes. And what I'm trying to do here is just to fit a linear combination of those 20,000 genes so that in the end, I only have like one score or two scores, two components. So I basically project my data that live in a space of size P into a space of size 2, for example. So this is what those components are telling you. And of course, the way, so you can do that with single value decomposition. The way it's calculated is that you want to maximize the variance of your data. And so when we say that, we mean that the variance of each component should be maximum. And what we also have are those loading vectors. Basically, they are the coefficient, the variable coefficient of each gene that you apply in your linear combination. So the higher that coefficient in absolute value, the most important that specific gene is to explain the variation in your data. And so there's been a very good, so the SVD would fit into a least square regression approach. And so you can use lasso to do variable selection there. So basically, you can implement lasso here on those loading vectors so that in the end, your components are not built on 20,000 genes. They only build on, say, 5 or 10 genes that contribute the most to the variation of your data. And so this is how we do variable selection. So this is PCA and sparse PCA. And I'm going to move to one of the approach we developed when you have multiple omic data sets. So here, for example, I have gene expression, I have proteomics, metabolomics, all measured on the same samples. But remember that they are produced by completely different technologies. So often people ask me, well, can't you just concatenate the whole thing and then you just do a PCA or whatever you want to do and you're done? They had completely different scales. And so using component-based approaches such as PLS or PCA actually project each data set into a space where they are comparable to each other. And so what we do is we also do matrix factorization. So similar to what I showed you before, we reduce the dimension of this data into a set of components. Here they're called latent variable in PLS, and you have the loading vector. But the way you do the statistical integration is that you actually ask for the component to be maximally covariant with each other because they're measured on the same sample. So you can do those calculations. And so basically what you're asking here is, can I find a component from the gene data set that is maximally covariant with the component from the proteomics data set that is also maximally covariant with the component from the metabolomics data sets. And so this is how you find those components, and this is how you project each data set into a small space. And each of those spaces are actually comparable to each other now. We don't have this problem of scalability, the fact that it was generated with from different technologies. And then you can do the lasso on those loading vectors. And so you end up with a very small selection of genes, proteins, metabolites that together explain the variance or the covariance between those data sets. And this is then what I give the biologist. I say, okay, from your multi-omic experiment, you ask me what is the major source of variation in your data, and I can tell you that these are the variables actually that you should have a look at. These are the ones that may make sense biologically. And the reason why biologists also love those approaches is because they're visual. So you can extract those components and you can plot them. So that corresponds to a projection of your data into a smaller subspace. So here I integrated 4 different types of omics data. So we have microRNA, kind of genes, mRNA, macroRNA, methylation data, proteomics data. So 4 data sets. And for each of them, I'm plotting component 1 and component 2. and the dots here represent the samples. So here, this is a very big public data set, probably about 150 samples. Usually you won't have that, but for testing the methods, we usually try to use bigger data sets. And each dot here represents the type of tumor that those patients with breast cancer have. So you have the basal tumors in blue, and then you have HER2 and luminal A and luminal B. And so if the method is working well at integrating those data, you should see very similar plots because you have managed to extract the covariance between those data sets. And so you can see it's not exactly the case. And you can start to understand what's going on also in the data. So for example, I would say to the biologist, well, okay, so the mRNA data set is pretty good. We can discriminate the basal samples from the other samples. I can tell you that luminal A and luminal B, based on the signature we selected as well, or based on the variables we selected, seem to be quite similar to each other. But if we look at the microRNA, then I can tell you that, well, actually, you know, this discrimination is not happening too much here. The data are probably very noisy. You should have a look at the microRNA we selected and tell us if it makes sense for you. So you can really start to dig more into what actually those data mean. So this is from the sample perspective. You can also have a look at the variables we selected, because remember, in those multivariate approaches, what we're really interested in are what are the correlations? What is the correlation structure between those variables? And so similar to before, where I was telling you we project the samples into a subspace that is spanned by the component, we can do the same for the variables. And I don't know if you've seen that before. I don't think they're very popular here. They're extremely popular in France because we're all fan of PCA. So I don't think there's, I think any students at the university knows what PCA is in France. It's just to tell you how popular that is. And so this is a very specific representation of a result of PCA. So what we do here is we have the components, and we calculate the correlation between the original variable. So this is the expression of one gene with the component. So the x-axis is my first component and the y-axis is my second component. This is how you project your variables into the space spanned by the component. And because it's a correlation, it's going to be within that correlation circle that has a radius of 1. And so you can do that for every variable you have. So here I'm just showing you 4 of them. And what is interesting is that, so basically you want to have a high correlation because that tells you, so that's a long arrow, because that tells you that those variables are important in defining the component. So those two, x1 and y1, if I project them onto the x-axis, the first component, I can see the correlation is pretty high. So x1 and y1 are actually very good at, contribute really well to define the component. And then you can look at the angle between those variables. So if the angle is small, so the cosine angle will represent the correlation between those variables. And so you can start to see that, oh, okay, x1 and y1 are actually highly correlated together. Well, yeah, that makes sense because they both explain, or they're both important to explain the component 1. And so they must probably explain similar information, similar variation in the data. On the contrary, x1 and y2, the angle is square, and so they're not correlated. And that makes sense because x1 contributes to component 1, and y2, if I project on the y-axis, contributes to component 2. And by definition, in principal component analysis, the components should be orthogonal. Okay, so if one variable explains one component, it shouldn't explain the other component. And the other thing that I tell biologists to look at are those obtuse angles, where basically it shows a negative correlation. So if the expression of that gene is is high in the disease sample, it might be low. The expression here of that gene would be low in the healthy samples. So you can imagine you calculate this for every variable you've selected in the method. And here I'm showing you the results when we integrated 2 data sets. So the orange data set and the blue data set. And you can see now, so I'm not showing any arrows anymore because it gets busy, but you can see there's a set of orange variables that are highly correlated together. They are negatively correlated with some blue genes. And so you can start to understand what's going on in your variable selection. So the reason, and so this is where you can start to visualize really the integration that is happening between different layers of biological information. So the reason why I'm spending so much time on this is because we're going to use that information to integrate time course omics data. So moving on now to the time course multi-omics. So you can see it that way. It's the same as before, the same issues, except that now we have multiple omics types. So imagine that now when the patient comes to the clinic, we can measure all these different things on them. Or you can represent it as those cubes, it's the same. And so the idea is that we can actually, again, reduce the dimension first using linear omics model splines. And so again, so basically you end up with a data set with a very small number of time points and a large number of variables for each omics. And then you can put them into any integration method I told you about. So you can do the PCA, you can do a PLS, except that this time, each variable here is actually a time profile. And so from there, we can start to understand which are the time profiles across time that explain the variation in the data, which are the gene expression profiles that are maximally correlated with metabolites and so on. So you have to imagine now that we are in a much more complicated problem where we have the time component as well. And so from there, you can start to basically extract those profiles and have a look at their correlation. So this is the last framework that we developed. It's called time omics. By the way, because I try to push my students to not use all these crazy acronyms when we have a name of a method. So often, time omics doesn't correspond to, it doesn't say that we use splines or anything, but at least biologists understand, they remember those names. And so the idea is basically using the LMMS. So you fit a spline and then you input them into the multivariate methods I was telling you about. And then you can identify the groups of variables that are correlated together. We use the sparse, so we use lasso, and so we provide also different parameters to decide how many variables we need to select, how many components we need in those models. And this is basically the main output is you have time on the x-axis, and this is the expression of all the omics variables that you have selected. And based on the fact that we know that they are positively correlated, negatively correlated, that they contribute to component 1 or component 2 and so on, you can then basically assign each of those profiles to different classes of pattern. And so you can see here we have 3 types of variables, the blue, orange, and the gray, and you can see that they are neatly correlated just by using the properties of PCA and PLS, basically. And so this is really interesting. I mean, I don't know if you find that interesting, but for a biologist, it's really interesting to see that there are different patterns that occur across time, depending on, the disease or the intervention that is happening, that there are some genes that behave the same as specific proteins. The expression decreases and then increases and so on. So this is actually really insightful for them. And they can't find that from the data they give me in the first place. And yeah, so this is just a summary of the framework. But I just wanted to emphasize that while, all the stats bit is happening at the bottom, where you use, either PCA, PLS, or multi-block PLS, there's a lot of work beforehand to make sure that the data are, they've been quality controlled, they've been filtered, they've been normalized. And I think the issue is that for every different technology you use, you have to use a very different normalization scaling approach. I don't know if you familiar with the term normalization in biology, but basically it's applying some sort of transformation so that you remove as much individual variation as possible from the data. And so for microbiome, you may do a specific process, and for metabolomics, you may do another process, and that's actually taking a lot of time. And so a lot of my students are surprised when I say, well, actually, you know what, the processing of these data is probably going to take us about 70% of the time. The methods we have, that's all good, but before we get there, we need to do all the normalization. And in order to know if it makes sense, if it was a good normalization, we need to analyze the whole thing and then have a look at the results and say, yeah, that makes sense, or no, why are we finding this result? Okay, go back to step one, normalization is not correct, and blah, blah. And so it's a very repetitive and often frustrating step that we have to go through. So I'm going to give you just two examples of this type of problem. One is actually from, it's a public data set. It's looking at the first 100 days of life of infants. And they were born either through C-section or just a natural birth. And so these are just the experimental design. So you have time on the x-axis, and these are just the dots to represent the sampling that has happened across time. So you can see there's only like 5 infants per delivery mode. And you can see it's densely sampled in the first probably 10 to 20 days. Probably when you can still give, you know, the fecal sample, the parents give the fecal sample, and then after that, it starts to spread. And so you can just use the framework I showed you before. You fit linear mixed model splines and you do a PCA. And this is what happens. So you have to deal with each group of infants separately. And these are the different types of time profiles you identify, some straight lines. So those are bacteria that just increase with time. And then you see some interesting things happening here where you may have a bacteria that after a bit of time, the abundance increases and then decreases and then increases again. And what was interesting is that when you look at the infants that were born via C-section, apart from the straight lines, the profiles look different. And we know that a vaginal birth actually brings a lot of microbiome to the infants, whereas for a C-section they don't. And so you can then start to see that there are different behaviors of the microbiome across time, depending on the way those infants were delivered. The other example I want to show you is with Olivier. So Olivier is a microbiologist, and he works in the digestion of biowaste. So what he does is that he has those bioreactors. They can be small, they can be industrial scale. He puts all the biowaste in there, and then he measures everything he can. So he measures how much gas it produces. He measures, what are the bacteria in there? What are the metabolites that are produced? And the idea is really to maximize the digestion of this waste. And so he has 3 little bioreactors that he looked across 150 days. So when I work with him, I have to tell him 2 years in advance. Do you have any experiment that's coming up in the next few years so that I can have a time course experiment there? And what I like is that everything's very controlled. You can control the pH, the temperature. There's things that are highly controllable. And so he would measure performance measure of the bioreactor, such as how much methane is actually coming out, and the production of carbon dioxide and so on. And then he did the microbiome experiment as well. You can see, so this is 90 taxa, so it's quite a small data set, but we only have 4 time points. Now he has more, but that was before I met him. And now I'm like, no, can you actually do a bit more time points? And then he measured also the metabolites. And so for that, because we have 2 omics, we have we have 3 different types of data. We have performance measure, we have the microbiome, and we have the metabolites. Then we did a multi-block integration PLS there. And I just showed, so this is how it looks like. Like if you just plot what you have, you can't see anything. I mean, So that's why I'm saying I do a lot of data mining on the data. And this is what you get in the end. That's the final result is I say, well, actually, you have about two different types of profiles, the one that go up and then kind of plateau after some time, and the one that goes down and then kind of plateau as well. And this is showing the expression of specific metabolites that were selected with the approach, the taxa, and then the performance measure. And so I give that all to him and he say, oh yeah, okay, no, I can see what's going on here. There's a lot of production of methane and carbon. And that's really shows us that the degradation process is happening. And so those are the different type of archaea that are involved in that process. And here it's like, well, actually those bacteria are eaten by another archaea and that produces a lot of biogas by doing that. Of course, I wouldn't be able to come up with this kind of interpretation. So this is why the interactions with the biologists are really important for those kind of data, because I told you how limited we are, statistically speaking. In the next 5 or 10 minutes, I'd like to talk about the fine print of those approaches, because there are many challenges we still need to face, and especially in microbiome data. So if you're interested in working in that field, it's a very exciting field because there's heaps of money that has been put into all these microbiome experiments. So we're generating a lot of data. For example, in France, I'm working with a facility that produces a lot of microbiome data, and they aim to sequence 100,000 French people in the next 5 years. And so we have about 5,000 samples coming next year, and they're all healthy people. And we have only some clinical information, geographical information, but basically they want to characterize what is a healthy microbiome in the French population, and is there any difference between different geographical sites, for example. But the statistical methods are lacking, and for a good reason. It's because the data are extremely complex. And so I would say as usual, but really as usual here, we've been really lagging behind data integration in data generation. So I would definitely welcome a bit more theoretical input from the community here. So the fine prints are, first, what we are measuring with those sequencing technologies, and that's the same if you look at RNA sequencing. We're measuring the relative abundance. We're actually not measuring what is really there because we don't know what is there. So the ground truth is the absolute counts so this is a given taxa across 2 individuals. So this is just an amount that you see. But in fact, we don't see that at all. What we get from the machine is the relative abundance. And it's a relative abundance because you only sample a finite amount of biomaterial, and then you sequence that. So you just hope it's representative, but you don't know. And it's all relative to what else you've sampled in that biomaterial. And so sometimes you have a lot of material, sometimes you have a small amount. This is what we call a library size. And so that matters when you compare across different time points, because you may say, well, actually in time point 2 for that individual, I didn't manage to sequence a lot of stuff here. But you don't know if it's because, well, that individual was sick, or if it's because, well, that's how the experiment went. And so you can see that, we have the relative abundance where we say, oh, this bacteria the abundance increases across time. But in fact, it doesn't. It's actually quite flat at the beginning and increases at the end. So and that goes back to the compositional issue of those data, where we don't know what actually the truth is there. And we're just trying to work out and trying to find patterns in the data, but we actually won't know. The good news is that we now have access to more precise experiments or technologies where we might be able to measure the absolute counts, the absolute abundance. So I'm trying to push my biologist colleagues to do those experiments for me, as well as do the old experiments, in order to see if the way we analyze the data, the way we processed it, is actually quite reflective of what is supposed to happen. And yeah, I mentioned compositional data. So this is a very well-known problem now in microbiome data analysis, is that because we're working with relative abundance, those data live in a simplex. And so there's been quite a bit of work done around how do we get out of the simplex. So Aitchison proposed different types of transformation. And one that we use is the centered log ratio transformation. It's quite common, but it's quite basic as well. And what is happening, and it's just a plot here of the CLR transformation of the abundance of one taxa for two individuals across time. And the CLR transformation is you divide by the geometric mean, but those means may not be the same from one individual to another time point. And so there's a lot, I think I know there's a lot of issues when you compare across time, because you ask yourself, well, is it really comparable? Am I looking at the abundance of this taxa from time 1 to time 2, and can I actually do this? So this is where I think we need much more theoretical input. But so far, my discussions with statisticians has not been quite successful. They all look at me and they say, "Oh yeah, it's a big problem." But I think it's a big challenge. And so there's, I don't know if you know David Lovell, but he's been fighting for compositional data analysis for quite some years now, because he was saying, oh, we've analyzed this data all wrong all the way. How could you even dare doing that? Because they're all compositional, you don't realize that there's spurious correlations in your results. And so he proposed a proportional distance. When you work with compositional data, you always have to divide by something in order to try to get out of the simplex. And so he proposed that approach where basically for each time profile each profile, you can divide the variances. And so we kind of use that post hoc. So we would do all our analysis, and then we would say, hang on, do our results make sense? Let's apply this measure at the end when we have all correlated profiles across time to see if really we manage to find that association now using a more compositional approach. But yeah, there's a lot of work, I think, that remains to be done in that space. And as you can see, it's becoming more and more complex. And I think if you are keen to work in microbiome data, what you need to remember is they are count data. They are extremely sparse. There's a high individual variability, and good luck when you have time course experiments, because it's becoming even worse. I mean, those characteristics are amplified. So just a summary of the different types of analysis that you can do in omics in general, but I focus mostly on microbiome. Differential analysis, I think we've done a lot there. I would say we've covered most of what could be covered. So I wouldn't spend too much time there. I think where things are starting to miss is when you start to want to cluster time profiles. And the network modeling, I think, especially in the time course experiment, is something we're working on, but so far no huge breakthrough. And so just to talk about how we disseminate our methods is through R packages. So Mixomics is the big one. For those of you who haven't heard about it, it's a very popular package that I started to develop at the end of my PhD. And it's interesting because it really pushed my career just with that single R package. But the way I designed it, I mean, because I have an engineering background, I think I designed it the way that would be appealing for users in terms of the outputs that we get. They're all component-based approaches. So people always see the sample plot. They always see this correlation circle plot. And they're like, oh, okay, I understand it once. for that method, how I should interpret those results. And now I understand how those interpretations should be similar when I apply other methods for other questions. And I think that's been really successful. And so now we're pushing more towards the longitudinal aspect with some other packages. And so those are the people I would like to thank. There's quite a few funding bodies. During lunch, we were talking about the classical funding bodies. But you also have things such as the Chan Zuckerberg Initiative. They love to have innovation and kind of blue sky research. So they send a lot of calls. It's highly competitive, but it's good money. And I'm also part now of the new Center for Excellence in Quantum Biotechnology, because I think what's really driving my research are the new cutting edge technologies. And so there's new microscopes coming in the next 7 years. And I think that's going to create additional statistical challenges. Thank you.
Yeah, thanks very much. Are there any questions?
I was interested in when you use the CLR transform, and I'm guessing those among significantly this why you didn't just use the raw proportions, like why you transform those with your spatial?
Because the proportions are compositional already. So that's the problem, is that by the way the data are generated are compositional.
Yeah.
Because the abundance of one taxa will depend on the abundance of the other taxa.
Yeah.
So that's why.
I would have thought like splines would have worked on those proportions like without having to do a CLR.
I still think we still in that compositional space.
Yeah.
That's why I'm trying to get out of that space using CLR. But and the good thing with CLR is also that the data seem to have a more Gaussian distribution. So that helps as well a lot. But yeah, basically, when you work with microbiome data, unless you have specific models that are, zero inflated, and there are some constraint regression models also that have been proposed, you need to, and that can take into account that compositionality in the model. If you can't, then you have to transform the data in some way. Yeah.
Some question for the mathematic law PLS. That is a
Yeah.
What's the difference from CCA canonical propagation analysis?
It's I think the idea is the same. You want to maximize, well, for CCA, you want to maximize the correlation between components, and PLS, you want to maximize the covariance between components, but the algorithm are actually quite different. So in CCA, you inverse the variance covariance matrix. And so of course, when you have a large data set, that doesn't work. In PLS, we use partial regressions. So we do, and I can show you more details later, but we do a regression on the components. We use the components as a surrogate of the other data sets so that we don't have to inverse matrices. So that makes it extremely efficient for big data.
So you care about the covariance system?
I think it's about the same, because basically you can maximize the correlation also in the PLS if you scale your components, right? So it's not so much a correlation covariance, it's more about how the algorithm solve the optimization criteria.
Okay.
I have more questions. Well, the talk was really great, so thank you very much. Maybe one of the questions I had was if you do any collaboration with I think it's called the One Binome Project, it's CSIRO.
I haven't done that.
Okay.
Yeah.
Good.
Yes, there's quite a few initiatives. That's why I said there's a lot of money, or at least to generate data, not for the analysis. And there's been quite a few big projects, including Bioplatform Australia and SARO, to try to I don't know why they all want to sequence the microbiome in different ways, but it's not necessarily well designed. So, but yeah, just so the one thing we developing now is actually something a bit different. It's a stabilized regression, where basically we're trying to account for the batch or the cohort effect in there. So we're asking, I know that my data are coming from different countries, or they're coming from different cohorts or different batches of sequencing. And what is the microbial signature that is actually robust across those different batches? So we found some very good results. I didn't think we could do that. But that's one big issue also in microbiome is batch effect. Or any cohort effect of any kind. And you'd be surprised, but biologists don't realize that. So they send me the data and I say, how many mice do you have per cage? And like, oh, what do you mean? I said, well, they will have the same microbiome in that cage. They stayed there for 6 weeks. So what do we expect? And so there's a lot of work as statistician to talk to them and say, look, I know you can't put 1 mouse per cage, but honestly, my experimental unit is going to be the cage. So, you know, we need to take that into account with you and help you to design the experiment the best way. And the problem with those consortia, like, you know, any microbiome, big microbiome consortia, is they don't really think about that. And so they end up and they say, oh, yeah, so we have that cohort from that country and we sequence it in that country. And then we have another cohort and we sequenced it in the other country, and they use different machines or they use, you know, different technical people to do that. And then you end up with those disparate experiments. And so really the work is how you integrate those different batches to find the common signal. So I find that quite interesting. There are a lot of public data sets out there also you can find.
Yeah, so Karen has talked, and when you're fitting like the mixed models and then using the results later on, then you get some really nice tight curves, but how do you propagate the uncertainties related to some of those?
Yeah, I mean, the only thing we know is that, you know, we take into account in the linear mixed model framework, we know that we take into account the individual variation and all these things, but we don't have any measure after that. That's why, because it's so limited from a statistical point of view. And that's why I keep saying, oh, you know, we go back to the biologists and we ask them, you know, does that make sense? Or do you think there's something weird in our results? Because that's only validation that we can have. And of course, you can simulate data and do those kind of things, and we do that in every paper we have. We have, you know, extensive simulation data to try to benchmark everything. We show it works, but then in real data, it's, yeah.
No more questions? All right, thank you very much for coming.